Hello everyone, welcome back to my channel Xgene and proteinX. In this tutorial video, I'm going to talk about overview of the ELISA. So I'll focus mainly the ELISA principle, but not the ELISA protocol. So my later video, I'll discuss about the ELISA protocol. So let's begin. So first you have to know that what is ELISA. So ELISA is the enzyme-linked immunosorbent assay. So why enzyme-linked? Because the secondary antibody that used in the ELISA, that is HRP conjugated, and this HRP is the enzyme. So this is why these techniques are known as the enzyme-linked immunosorbent assay. So ELISA are a type of immunoassay that are commonly used to quantify levels of a specific target within a sample. So specific target is either antigen or antibody, and the sample routinely used, there are different kind of the samples like serum, plasma, cell culture supernatant, cell lysates, saliva, tissue lysates, or urine. And different stages of the cancer it also detected by the ELISA technique. And additionally, also the human chorionic gonadotropin or HCG in urine, which is higher in pregnant women, that also be detected by the ELISA. So next is the principle of the ELISA technique. So usually the ELISA technique it performed in the 96-well plate. So first the antigen or antibody need to coat it in this 96-well plate by using the coating buffer. So after coating the antigen, the primary antibody incubation required. So this primary antibody, this is very much specific against this antigen. So this primary antibody it bind against this antigen, and then the secondary antibody incubation. So this secondary antibody it bind this primary antibody, but the secondary antibody, this is the HRP conjugated, that is the enzyme. So this is why this process is known as the enzyme linked immunosorbent assay. So next is the addition of the substrate A that contain hydrogen peroxide, and subsequently need to add the substrate B that contain TMB. So these both are the substrate for this HRP enzyme, and then the one blue color develops. So this blue color intensity it's proportional to the HRP activity that the primary antibody that bind with the antigen. So if the higher amount of the primary antibody that bind with the antigen, that means the higher amount of the secondary antibody that bind the target primary antibody. So it means that the HRP activity will be higher in this case. So in this case, the higher blue color intensity. But this blue color, it's gradually increase unless this reaction stops. So this is why this reaction need to be stopped by using the stop solution. So this stop solution is the sulfuric acid, and then the yellow color produced. So this yellow color is very much stable, and this yellow color it read under 450 nanometer wavelength. So next we need to know the HRP activity. So HRP activity, this is an enzyme and it derived from the root extract of the horseradish plant, and that scientific name is the Amoresia rusticana. So when we add the secondary antibody, that is the HRP conjugated, and then it react with the hydrogen peroxide that we add as a substrate A. So here it forms a complex, that is the HRP hydrogen peroxide complex. So the initiation of an HRP enzyme mediated color developmental process for signal generation is dependent upon the presence of hydrogen peroxide. So hydrogen peroxide, how it works? So if we analyze, the hydrogen peroxide contain two OH group, and this O and O this link together by covalent bond, and this covalent bond is very much weaker compared to the other covalent bond. So normally this covalent bond for disruption of this covalent bond is required around 45 kilocalorie per mole, whereas for the CO and NO it needs around 84 kilocalorie per mole and 53 kilocalorie per mole respectively. So if you compare, you can understand that this is very much weaker. So this is why this low O and O bond energy factor enables hydrogen peroxide to act as a potent 2-electron electrophilic oxidizing agent. So as you know that hydrogen peroxide is very much strong oxidizing agent. So here the HRP structure, and the HRP structure contain on iron group or heme group. So this iron actually it's the native ferric form. It's normally is formed as a ferric form, right? So this ferric ion it donates 2 electron to the hydrogen peroxide, because as I mentioned earlier that hydrogen peroxide, this is very much strong oxidizing agent, so it accepts electron from the another compound. So here the hydrogen peroxide it accepts electron from the ferric ion, so then it forms Fe4 positive, and the TMB that is we add as a substrate B. This TMB is usually colorless, but this TMB is the main function of the TMB is a reducing agent, so it donates electron to this compound. So this compound already lose electron that donated to the hydrogen peroxide. So then this electron that is compensated by the TMB by donating the electron to this compound. So while the TMB it donates electron, then it changes the color, the blue byproduct color, and the maximum absorbance of this color is around 605 nanometer. So this blue color is not stable because the intensity it increase till the reaction end, but reaction need to be stopped by adding sulfuric acid. So when the sulfuric acid add here, and then yellow color developed. So this yellow stabilizing the color development to enable accurate measurement of the intensity at 450 nanometer wavelength. So next is the different substrate of the ELISA. So there are different kind of the substrate of the ELISA of HRP. So these are the substrate list of this HRP other than the TMB. So in the ELISA only need to use the TMB. Why? Because TMB has the highest sensitivity on HRP compared to the other substrate. So this is why TMB used in the ELISA. And here all of the other kind of this HRP substrate, their generic name and the scientific names. So I hope this video will be helpful. If you like this video, kindly hit the like button, share it, and please subscribe my channel. If you have any queries, kindly write in the comment section. Thanks.